Okay, good. First of all, thank you all for coming to today's lecture. As you know, this is a special sort of lecture. It's the distinguished postdoctoral lecture, and it's the highlight of the UCLA Neuroscience calendar, as you all know. And it's actually a very unenviable task to choose one postdoc per year to receive this distinction. And it's a very difficult task because the postdoc pool at UCLA is very deep, and it's very large, and there's a tremendous amount of talent here. And so even though only one person can be chosen, I think it's fair to say that all postdocs at UCLA are highly appreciated, and please carry on all your hard work. But today, the recipient is Jason Stein. And before I hand over to Dan, who's going to say a few words about Jason, I wanted to say something about the lecture. So if you look back, the lecture's been running for over 10 years now, I think. And if you look back at past recipients, they've nearly all gone on to land faculty jobs. And so this is a very good predictor for future success, if of course you consider a faculty job as success. And about 2 years ago, the lecture was changed, the title was changed to the Arnold Scheibel Distinguished Lecture. And the reason that was done was to honor one of our former neuroscientists who's done tremendous work at UCLA in terms of leadership and service, but also tremendous work in terms of neuroscience, scholarly achievements. So Dr. Scheibel's not here, but I understand he's watching via the net. And so I just want to say hello, thank you, and on behalf of all the students, postdocs and faculty, we salute you for your achievements and your contributions to UCLA Neuroscience. And with that, I'm going to hand over to Dan now.
I just want to second that mention of Dr. Scheibel, who is really one of the great neuroanatomists of the last century, really phenomenal neuroscientist, and he's very, very well known for his course in neuroanatomy, which is the best I've seen anywhere. Just remarkable. And I remember when I'd have some postdoc geneticist in my lab, and I said, well, you got to learn about neuroscience this year. And they would go take his course and just say, that's the most amazing lecture I ever heard, kind of every week. So an amazing, we're lucky to have that. Searching for words in the old age, we're lucky to have that kind of hardly different resource. Yeah, heritage, thank you. Heritage. So I have one thing I just wanted to quickly show here as I introduce Jason. So 12 years and 4 months ago, Jason graduated from Northmont High School in Clayton, Ohio. Valedictorian, of course. That's 2002. And since then, it's been all downhill. No, since then, obviously, he's done remarkable things, but I was looking, and the featured administrator this month is the principal, George Karras. When I called him, he hadn't been around. He was in the elementary school, so he didn't really remember Jason. But I was a little disappointed because today the daily announcements don't really include this lecture, and their distinguished alum senior assembly auditions will take place, Mr. McMongle, 7th period, blah, blah. All Wrestlers Tuesday in the pit. And the marching band did very well as well. So, what can I say? But it really is a great pleasure to introduce Jason with his beginnings in Ohio, and he still is the major supporter of Ohio in our laboratory. Following that, he went to Northwestern University, where he majored in integrated sciences. He was Phi Beta Kappa, and very interestingly, he was a member of, I didn't even know, he was a member of a fraternity. So I was thinking, all right, party animal. No, it was actually, it's a physics fraternity. Sigma Phi Sigma. So he was inducted into that. Let's see what else. I can't even read my own writing, which is a part of the, but anyway, here he is really Jason Stein. At UCLA, he did his PhD, really a distinguished PhD with Paul Thompson, on the genetic influences on brain structure. And it was really remarkable because he wrote a Pathway to Independence Award, right after he came to my lab, and he was one of the finalists. And the reason he got shot down in that is because of a guy who will remain nameless, who's no longer, who is a distinguished geneticist in academia from Princeton, involved in linkage, some of you may know who that is, who basically said, this study that you want to do is not doable, it's not going to work, it's all garbage. And then the next year he published it as first author in Nature Genetics. So that was, so he had over 30 papers when he finished his PhD, and now I think he's over 40. He did the, he was, as a graduate student, he won the Samuel Idison student lecture in 2011. He's been an ARC scholar, a neurobehavior genetics fellow as well. He's really just done remarkable work in the lab, and in talking with him, he had been doing, just to give you a sense of where his head's at, he really knew how to do imaging analysis, knew quite a bit about statistics and statistical genetics, and had put them together in his thesis. And really, I think could have gotten a job doing imaging genetics and neuroimaging analysis after his PhD, but decided, I want to know how genetic variants influence at a mechanistic level. I can see that something might vary a hippocampus or a caudate, but I want to know what it does in a cell and how it actually leads to disease. So he has a burning passion about science and wanting to make a dent in neuropsychiatric diseases. And so he came and has been plugging away at the bench, which he's going to talk about. But he is, he is a kind of idiosyncratic guy. He has some idiosyncratic dress. We wear a lot of protective gear in our laboratory, a lot of toxins. So there it is. But I should say that he's also revered by people in the laboratory, and is even so well revered that there's a Halloween costume some people wear, and there is one of the other lab members, hey John, being Jason, for Halloween. So he was hey John, yeah. I didn't want to go there. So now back to, anyway, with that, it's really, that's the wrong one probably. Let's see, Scheibel. There we go. Let me get him started. But it is all kidding aside, he's able, to really balance his work with his life. That means 99% work, 1% life, like the rest of us. But he's really been remarkably successful. He's a great person to have in the lab, and it's really a great pleasure and a great honor, really, to have him in my laboratory and to be able to introduce him today.
Thanks very much, Dan. It's always dangerous to have Dan to introduce you. Thank you for that introduction. First, also, I'd like to thank Dr. Scheibel. So I am a proud graduate of Dr. Scheibel's neuroanatomy class. This is all of us graduating in 2008. You can see all of us, this is actually at Dr. Scheibel's house. Oh yeah, there we go. So I don't know if Dr. Scheibel, you can see this, but this is all at your house back in the year 2008, right after our exam, when we survived the exam barely and really learned a lot through your class. We all came over to his house, and we talked about stories of neuroscience in the days past and just hung out. It was really a great time. You can see Dr. Scheibel here looking very dapper with all the neuroscience ladies surrounding him. You can also see me in a different haircut there. I went with the fro back then, but now more close cropped. Okay, so thank you guys so much for coming, and thank you for this honor to be able to speak to you on Now Some Science. So there have been a stream of genome-wide studies trying to link specific genetic variants to risk for diseases. This has been particularly crucial in neuropsychiatric diseases which have previously had no causal basis to them. So finding these specific genetic factors gives a causal basis to these disorders. Here is just a toy example. You can see that the T allele here is overrepresented in patients with schizophrenia, whereas the A allele is overrepresented or is more often in controls. When we do this in just a few subjects, it doesn't really give us much information, but when we do this on the order of 10,000 subjects, this is able to give us for the first time genetic causes for a disease. So we can say that the T allele of this SNP is associated with schizophrenia or autism, or whatever disorder we're interested in. So this research has been going on for the past few years. What this research doesn't give yet is a mechanism by which a single base pair change or any kind of genetic variation change creates risk for a disease. So to go after this mechanism, there are many model systems that we can use. And those can be parameterized along 3 axes for how good of a model system they are. One is mechanistic insight, so how much can we modify the genetics of this particular model system. Another is throughput, how quickly can we create this model system. And a third is human relevance, how relevant is the system to the actual human brain. So mouse models that most people are familiar with have really high mechanistic insight. We can modify their genetics, we can run a lot of different experiments on them, we can do really well-controlled studies. However, they have low throughput and low human relevance. They're mouse, they're not human. What is new in the field is induced pluripotent stem cells and primary human neuroprogenitor cells that seem to be high on all 3 of these axes. So they have high throughput, we can generate a lot of these different neural stem cells. They have high mechanistic insight, we can modify their genetics and run well-controlled experiments on them. And they are human, so they theoretically have high human relevance. So neural stem cells represent an excellent model system for modeling genetic effects on brain structure or risk for disease. However, what we really don't know very well is this y-axis here, this human relevance axis. How well do they model in vivo development? So neural stem cells can be derived from either embryonic stem cells, induced pluripotent stem cells, or directly from fetal tissue. And I'm going to talk mostly about derived directly from fetal tissue. Neural stem cells are one of the few ways we can study the living cells of the human brain. Since neurons don't divide, there's not really a great way that we can study these other than neural stem cells or biopsies from patients. We can genetically modify these cells to be similar to a patient with a disease, or we can take cells from a diseased patient and grow them that have the same genetic basis as the patient. And lastly, theoretically, these can be used for high throughput drug screens, which will be extremely useful for a field that has seen no new medications developed in the past 50 years or so. However, neural stem cells have a great degree of limitations. So cells mature extremely slowly, similar to the in vivo time frame. So it probably takes about 9 months to form a mature neuron, which is just about 9 months of gestation. So a mature neuron in a dish takes about the same time as in vivo. The regional circuitry is lost, obviously, if you just have a 2D culture. Cellular identity is not easily controlled, and cultures are often heterogeneous mixtures of multiple cell types from multiple regions. So the central question we want to ask is, again, cells are grown in a dish. How well do they model the development of the brain? So the way our lab mainly studies this question is using primary human neuroprogenitors. So primary human neuroprogenitors are derived from fetal cortex. So we take fetal cortex, dissociate it into a single cell suspension, generate neurospheres using mitogens, establish a monolayer culture, and then we remove the mitogens that we were interested in using, add BDNF and NT3 to induce neuronal differentiation. We differentiate the neurons for 1 to 12 weeks, and then our outcome measures that we use are immunocytochemistry or transcriptomic analyses. I'm going to talk a lot about the transcriptomic analyses, but first I'll show you some of the immunocytochemistry. So the most common way of determining if the cells do what you want them to do, if they form the type of region you're most interested in, is to use immunocytochemistry for markers of interest. So one of the markers that we study, or that is commonly used for telencephalic fate, is FOXG1. Markers that are used for radioglial fate are SOX2 and PAX6, and ASCL1 is a marker of a GABAergic progenitor. So we can look at all of our cells in the dish and determine how many of them express each of these different cell type markers. So what we can see is that 100% of the cells express FOXG1, implying they're dorsal telencephalic, which makes sense. We derive them from fetal cortex. And then almost 100% of the cells express both SOX2 and PAX6, implying they are consistent with the fact that they're radioglia. And about 36% of them are dividing at any one time. And also, this is just the first of this, but Luis and I, who's another postdoc in the lab, did all of this work together, so this is just the first Luis face you'll see, but he is essential to all of these projects that we're talking about here. So the next thing is that we want to see, okay, we have primary human neural progenitors, we know that they're telencephalic in fate, do they differentiate into neurons? And you can see here, through time, from week 1 to 4 to 8, you can see an increase in MAP2 staining, which implies that there's more neurons being formed. Here you can see about 40% of our neurons, 40% of the cells formed are neurons. You can see also a decrease in nestin, or neural progenitor staining, so the cells are turning from neural progenitors into neurons. You can also see a decrease in dividing cells because the neurons are not dividing, only the progenitors are dividing. We can also see that differentiating neural progenitors exhibit stereotypical morphology. So you can see at the beginning they form these stereotypical bipolar type of neurons. After 5 weeks in a dish, they start branching out and forming axons. Here you can see an axon, and here are the different processes that come off of the neuron. And here you can see even more processes being formed at 12 weeks and still an axon. And we can quantify this and see almost a linear increase in total dendrite length and average number of branches. So you can see that the cells are actually differentiating and showing stereotypical neuronal morphology. One more question we can ask is, do these cells follow the stereotypical processes that happen in the development of the human brain? So we know that lower layer neurons are formed first and then upper layer neurons. So do our cells follow the same type of process? Well, CTIP2 is a lower layer neuron marker, and POU3F2, or brain2, is an upper layer neuron marker. And so we can see that at early differentiation times, CTIP2 and MAP2, so this is, these are lower layer neurons, there's more lower layer neurons than there are upper layer neurons, as shown here. And similarly, expression trajectories are similar to in vivo cortex. So this is the in vivo cortex here, and this is our in vitro culture. You can see that CTIP2 comes on before POU3F2. And similarly in our culture, CTIP2 comes on before POU3F2. So this gives us greater confidence that what we're modeling in a dish is similar to what's going on in the human brain. However, what I've just shown you is basically where most studies stop in sort of characterizing their cultures. There are some limitations, though, to doing these marker-type studies. So the first limitation is that markers are often only specific given a neuroanatomical context. That means that we know that this marker is a marker of radioglia for cortex, but it's also maybe a marker for cerebellum. So PAX6 is an example of this, where it's a well-known marker for radioglia in the cortex, but it's also a marker for radioglia in the cerebellum. Also, though markers for regional identity exist, those markers for maturity are less well known. So if we want to judge the age of our cells, we have a difficult time doing that with markers. And finally, markers don't really give us an unbiased genome-wide representation of what's going on in the brain. So here you can see the lack of marker specificity sort of visually demonstrated. So what I'm showing here is expression shown by the color of this heat map. On the x-axis you can see different genes, and these genes are commonly used literature markers for different regions. So for example, for telencephalon you can see FOXG1, which has high expression in the cortex, the striatum, and the hippocampus, so all the telencephalic areas. So this is a pretty good marker. OTX2, however, is used commonly as a telencephalic marker, however it doesn't have such good expression marking telencephalon. PAX6 similarly has high expression in the cerebellum, you'll see, and a little bit lower expression in the cortex. So this is just motivation for taking a genome-wide approach that I'm going to go through now. So our goal was to generate methods to match the transcriptome from in vitro cultures to large transcriptomic in vivo datasets. So we used 2 in vivo datasets, and one of them is called the Spatiotemporal Transcriptomic Atlas of the Human Brain. This is a really fantastic data set where 56 postmortem brains were taken from 4 post-conception weeks all the way up to 60 years old, so they cover the duration of the human lifespan. They were also from multiple different regions of the brain, including both cortical areas, as you can see here, and subcortical areas, as you can see here. So this is a great data set because it allows us to match not only different time periods of brain development, but also different regions of brain development. Also, importantly, the entire cortical wall was taken for samples from 4 post-conception weeks up to 10 post-conception weeks. So that entire cortical wall contains progenitors. Whereas for the rest of those time periods, the cortical plate and subplate were taken, or for adults, the cortical gyrus, including some white matter, was taken. Okay, so that's one of the data sets that we're going to use as our in vivo standard. Another data set that we'll use as our in vivo standard is a laminar atlas of fetal brain development from the Allen Institute. So in this, they took 4 brains from 15 to 21 post-conception weeks. They flash froze these brains and sectioned them, and then performed laser capture microdissection to get the specific lamina of the human fetal cortex. So here you can see the lamina. Here you can see the lamina outlined, and here you can see the lamina after they were removed by laser capture microdissection. These lamina were then RNA extracted, cells from the lamina, RNA extracted, and then run on custom Agilent chips. Here you can see a zoomed-in view of the different layers of the cortex. Again, something we learned in Scheibel's class all those years ago. Just to zoom in there a little bit more. So these are the different layers of the developing human cortex. The different layers of the cortex contain different cells. The ventricular zone, subventricular zone, and outer subventricular zone contain the progenitors. They contain the cells that form the rest of that form the neurons and the glia of the cortex. So these are the radioglia, the outer subventricular zone progenitors, and the intermediate progenitors. Those cells form neurons that then migrate up through the intermediate zone and into the subplate, cortical plate, and marginal zone. So the neurons are up here, and all of the germinal layers are down here. Okay, so now we have 2 really great in vivo data sets. We want to develop methods so we can understand how well we can model the brain in a dish. So answering that question involves answering a few sub-questions. First, what regional and laminar identity do we model? So here you can see the different regions of the brain and the different lamina. We want to know what developmental time we're modeling. So are we going from the embryo all the way up to 60-year-old neurons in a dish? Well, it's hard to tell just by looking at the dish. We're trying to do that through transcriptomic matching. What functional processes are being modeled? We know that there's some stereotypical functional processes that happen. First neurogenesis, deep layer neurogenesis, then upper layer, migration, and finally gliogenesis. Which of those processes are being modeled in the dish? And finally, there's a whole lot of different systems for making, a whole lot of different in vitro systems and different culture protocols too. The field really has not settled on one as being the best. This is the best protocol or this is the best system. So those systems are fetal-derived tissue, somatic-derived tissue as iPSCs, embryonic stem cells, and even neuroblastoma cell lines. These are often used. These are cancer cell lines. So which cell system is best, and are there differences between cell systems? Which best models the in vivo development? These are all questions that we want to answer. Okay, so to answer this question, first we developed a technique called transition mapping. And in this technique, what we do is first create a differential expression gene list between week 12 differentiated cultures versus week 1 differentiated cultures. We then create a ranked list where we put all of the most upregulated genes on one side and the most downregulated genes on another side. We then do the same with the in vivo data. So we compare 2 periods of human cortical development from period 1 to period 2, for example. Then we create a differential expression list, rank them by their most upregulated versus most downregulated. After that, we try to overlap the 2 lists. So we take the first 200, say, genes from the upregulated in vitro list and the first 200 genes from the upregulated in vivo list. We determine how many of those overlap, and we can assign a significance to that overlap. Is that overlap more significant than we would expect by chance? After we do this, we can repeat this and get a heat map. Very colorful heat map. Those heat maps show genes that are co-upregulated together in the bottom left-hand corner and co-downregulated together in the top right-hand corner. So we can perform this for any 2 time periods, and we can do this serially, as you can see here. We get these very colorful maps that show significance for how much there is overlap between between the in vitro data and the in vivo data. This gives us both a significance estimation for how much overlap there is, and tells us which time period has the most overlap. So what we're seeing is that we're seeing a highly significant overlap between neuroprogenitor differentiation and the development of the in vivo cortex. And we're seeing a high overlap at stage 1 versus stage 6. So stage 6 is 19 to 24 post-conception weeks. So after differentiating these cells for 12 weeks, we get neural cells that pretty closely match 19 to 24 post-conception weeks, so late fetal development. So after we do this, we can ask the question, which of those genes are co-upregulated? Do those co-upregulated genes have any kind of known pathways associated with them? So we took all of those genes that are co-upregulated at the most significant points in the maps shown previously, and then put them into a Go ontology to try to see if there's any sort of pathways that are enriched. And we saw synaptic pathways and neurogenesis pathways enriched as being co-upregulated. And here you can see some of the genes, well-known genes, PSD95, SHANK2, and synapsin1, have very similar trajectories to in vivo patterns. Similarly, we can do the same with co-downregulated. So we can look at all the genes that overlap that are on the co-downregulated side, and we can see that cell cycle pathways are most affected here. And you can see cell cycle genes are downregulated in here. So this is as expected. Neurogenesis is turning on, mitosis is shutting down. So this is really what we expected to see here. The next thing we can do is try to see if we can use this transition mapping program to see if we can observe a progressive maturation. So we have different time periods that we sampled the human neural progenitors at. So we can compare 1 versus 4 weeks to using this transition mapping approach. We can then do the same for 4 versus 8 and 8 versus 12. And here you can see that the transition mapping shows a progressive maturation as you increase differentiation time. Again, this is really interesting to see because it allows you to see where your cells are in time. We wanted to do this not only with temporal mapping, but also to do this with laminar mapping. So we wanted to see how well our differentially expressed between 2 time periods maps differential expression between different lamina of the developing fetal cortex. So you can see here that the most overlap is found between ventricular zone and subventricular zone, which contain the progenitors, and subplate and cortical plate. This is again as expected and gives us greater confidence in the method. So that describes one of the techniques that we use, and it's called transition mapping. Transition mapping provides a genome-wide, unbiased method to evaluate how similar in vitro cultures are to in vivo, what level of maturity is reached after different differentiation times, and what laminar transition is best modeled after differentiation times. There's a caveat to this analysis method, though, and that it needs multiple samples for evaluation. We ran a differential expression in this analysis, so we need at least 3 samples at the 2 different time periods. So now we ask the question, can we do this at the individual level? Can we do individual sample prediction? So to do this, we used a machine learning algorithm, a multi-label, multi-class machine learning algorithm. We fed in the spatiotemporal transcriptomic atlas, all 1,340 samples, to train our algorithm. We then input independent in vivo data sets where we knew both the temporal and spatial criteria for each of those samples, and saw how well this machine learning algorithm was able to classify those new independent samples. And so what you get out are these heat maps. These give the predicted probability of belonging to a certain developmental time period and the predicted probability of belonging to a certain region. We then evaluated our accuracy and saw that it was sufficient, so then we can use our in vitro data, put it into the algorithm, and do the same thing. So now I'm going to try to prove to you that this actually works. So here are all the in vivo data sets we input into the algorithm. So here you can see 4 data sets. So there's 1, 2, 2, 3, 4 data sets that we input into the algorithm to test the accuracy. First, I want to draw you to the regional identity. So the true sample identity here was cortex, and you can see that they accurately predicted cortex. The true sample identity here was cerebellum, and you can see it accurately predicted cerebellum. More difficult is to predict the developmental time period of each of these in vivo data sets. So we knew which age that the postmortem brains came from, and we ordered them by greater age. So you can see that as age gets greater, there's greater matching at older time periods. And so here you can see, again, older matching at older time periods. Overall, age matching was not perfect, but using plus minus 1 from the accuracy level, we get about 84% to you can see in others, about 100% accuracy. So we did this, importantly, not just in one array, but across multiple types of arrays, both Illumina and Affymetrix arrays, and in multiple types of Affymetrix arrays. So we think this algorithm is robust to different types of arrays. It's robust to different time periods. Here you can see fetal development, and here you can see old age, and it's robust to different regions. So we feel that this validated our algorithm. And just to pause one more time and to talk about Dr. Scheibel. So I think after I just blew your face off with this, the massive amount of color in the previous slide, I want to say that the importance of coloring to neuroscience research was taught to me by Dr. Scheibel using the Human Brain Coloring Book. In this book, for those of you who haven't taken the course, what you do is you color each of the different regions of the brain, and this way you learn the anatomy of the brain. It's an amazing book. Everyone should get it. Okay, next science. So primary human neuroprogenitor cells were then input into the algorithm that we previously validated. And we did this for multiple different samples of different primary human neuroprogenitor cells. So each of these columns represents a different sample, and these are from week 1, week 4, week 8, and week 12. Here you can see at the top that there's a progressive maturation observed across different differentiation weeks. This is cool because you can actually see the maturation of the cells just by looking at their transcriptome. So we can see that the cells match at the earliest time points probably most to stage 1, which is similar to what we previously found, and match most at the later time points to stages 5 and 6, which is again what we found by the transition mapping approach. Overall, regional identity was strongly cortical for the most part. So this is good again because our data set in an unbiased way again matches the cortex. There is a caveat though to using this context algorithm, and that caveat is that each sample must be classified into a category. So even if you put in, for example, lung tissue into this, which doesn't belong in any of the categories, it will classify it as one of those categories because it must. So because each sample must be classified in one of the categories, we wanted to give a criteria beyond which you can use the algorithm and below which you should not use the algorithm. So we did this by using the rank-rank tests and judging the region accuracy based on different levels of rank-rank tests from the transition mapping. After doing this, we found that a negative log 10 p-value of this level, which is where our HNPs were, was sufficient to get very high levels of accuracy for the context algorithm. This was for both regional identity and for developmental period. So we believe that our cells were able to be classified well by this context algorithm. However, there is a caveat to the future use of the algorithm. Okay, so context is the second tool that I am going to talk about of 3. And context provides a genome-wide, unbiased method for individual sample prediction of both regional and temporal identity. That means you can throw in your differentiated neural stem cells into this, and it will tell you where they belong. It was validated on multiple transcriptomic microarray platforms and proved to be useful for in vitro assays. There are caveats though, as I discussed. They will always assign a regional and temporal identity, even if none match. And as of right now, it does not yet give the genes driving the recognition, but that's something we're working on. Those genes that drive the recognition might be very useful for predicting genes which can determine regional identity or determine maturity. Okay, so those 2 methods made me very happy because as a bioinformatics, you want to judge time and space. It's great. It's just like being, being a prediction. but what we didn't really learn is about the biology, right? So we didn't really learn new information about how much different biological processes were conserved. So to pursue that, we used clustering of genes into pathways via weighted gene co-expression network analysis. And many of you are probably familiar with this and have seen this slide multiple times. But what this does is it groups genes that have similar expression patterns across samples into functionally related modules. And those modules are often enriched in genes known to be involved in some sort of process. So using this, we can group genes in vivo, in the in vivo cortex, into different modules and see how well they're related to different processes known to happen in the in vivo brain. So what are the processes known to happen in the in vivo brain? Well, we have these neural stem cells. These neural stem cells divide to form neurons that migrate up along the radioglia. They also form intermediate progenitor cells, which then form neurons that again migrate up to the radioglia cell, and then after a while they form glia, and those work as well. So what we tried to do was create a network of genes interacting on the in vivo cortex, and try to see which of the modules clustered in different known histogenetic processes. So for example, neurogenesis and differentiation, neuronal migration, axonogenesis, programmed cell death. For each of these, we looked to see if modules were enriched in genes known to be involved in those processes. So here you can see some of the modules and their trajectories plotted over time. The modules have these arbitrary colors, which don't really mean anything, but just are a signifying criteria. So you can see, for example, the magenta module has highly increased, highly enriched genes related to cell cycle. And the genes involved are cell cycle checkpoint kinase kind of genes. You can see that those genes are have high expression at early time periods and low expression at later time periods, consistent with differentiation. Similarly, you can see the salmon module and the green module here are enriched in synaptic genes, and they have increasing expression trajectories over time. So again, WGCNA allows us to separate all of these different histogenic processes, and then we can use what's called module preservation analysis to see how well our in vitro data set fits this in vivo defined network. And so we can judge this using this module preservation criteria here, determined by this Z summary score. You can see that most modules, many modules I should say, are highly preserved many of the modules defined in vivo are highly preserved in vitro, and that 86% of genes are within replicable modules that are preserved at a Z summary greater than 4. So again, showing that multiple modules are preserved and multiple different processes of the in vivo cortex are being preserved in vitro. Next, we can try to see if disease-associated genes are enriched in any of these preserved modules. If they are enriched in these preserved modules, then we can study those specific disease processes in our differentiating primary human neuroprogenitor cells. So I'm going to focus on these rare de novo variants from autism spectrum disorder, the ones that just disrupt proteins in the probands. So they were highly enriched in this midnight blue module. This midnight blue module, as shown here, is involved with chromatin organization. And you can see that the genes specifically affected in autism, or the genes that have these rare de novo mutations in autism, seem to have similar network properties in vivo and in vitro. So you can see this on the x-axis here. This is the in vivo KME, which is the correlation between a module's expression and the module eigen gene. It determines the network hubness of that gene. And this is the in vitro equivalent. The red line displays the y equals x, so the equivalency between the two. And you can see that most of the genes that are affected in autism cluster very close to this line, implying that they're highly conserved in vivo and they're highly conserved in vitro. You can also see their trajectories for each of the different genes that are affected in autism. And you can see that their trajectories are highly similar to the in vivo trajectories here. So this gives us confidence that we can study these particular type of processes in primary human neuroprogenitor cells, that these are a good model for us to use. Also, rare de novo variants are enriched in genes associated with fetal development, as determined by 2 papers, both in autism here and in schizophrenia. So evidence recently is pointing to both the cortex and fetal development as being time periods crucial for both autism and schizophrenia. Our primary human neuroprogenitors, by this analysis, seem to have that exact time period being modeled. So again, it seems that our primary human neuroprogenitors are going to be good models for studying autism and schizophrenia in the dish. Okay, so this is the third method, third of 3 methods that we went over to try to evaluate neural stem cells, how well neural stem cells model in vivo development. So module preservation provides a genome-wide unbiased method for evaluating whether major histogenetic processes defined in vivo are conserved in vitro. It allows greater biological interpretability than the other 2 methods because you can actually say what these different modules do. However, the caveats again are that you need multiple samples for evaluation at different time periods. Okay, so I went over 3 different methods for comparing these in vitro systems to in vivo development. Now what we want to do is feed as many different data sets as we can into this algorithm to try to see how well different data sets match in vivo development and how much variability there exists. So we fed in one data set. This is an iPSC data set of neural progenitors and neurons differentiated. And we see that on the same scale as the HNPs plotted before, we see much lower matching to in vivo cortical developmental transitions, much lower matching to laminar transitions, highly varied regional identity, and really not very much difference in the temporal identity. We can further quantify this by comparing different overlap maps. So here you can see the primary human neuroprogenitors on the same scale as the iPSCs. You can see that if we statistically compare these 2 maps, we see a highly significantly different, highly significant difference between these 2 maps, where there's much stronger matching in the PHNPCs. And because this is in the lower right-hand corner, which is involved in neurogenesis, this highlights its differences in overlap for neurogenesis genes. Okay, so then what we did was we again tried to feed as many data sets as we could. So we called up our friends at different labs around the country, all doing iPSC work, and asked them to send us their RNA so we could feed them into this algorithm from iPSC-derived data sets. Here you can see there's massively different matching based on different iPSC data sets to in vivo development. You can see that some of them have pretty consistent cortical identity, whereas others have not so consistent cortical identity, even with strong transition mapping. So this really, I think, is a wake-up to the field that there's great differences between different labs in these iPSCs, and that your culture protocols matter a lot for determining how your iPSCs differentiate into neurons. We also did this with other systems, including embryonic stem cells and neuroblastoma-derived stem cells. You can see that embryonic stem cells seem to match in vivo development pretty well, whereas SY5Y cells, these are neuroblastoma cells, don't match in vivo development very well, which is kind of expected. But again, implying that heterogeneity between different model systems is very important. Okay, so after all those analyses, we can try to answer this question: how well can we model the brain in a dish? So, what regional and laminar identity were we modeling? Well, for primary human neuroprogenitors, we were modeling cortex pretty well, and we were modeling the transition between ventricular zone and subventricular zone to subplate and cortical plate. The developmental time period that we were modeling is really embryonic development to fetal development. So we are not reaching late-stage neurons of adult or old age. What functional processes are being modeled? Well, we model neurogenesis, neuronal migration, axonogenesis, programmed cell death, and synaptogenesis. We don't really get up to gliogenesis or myelination. And finally, what differences exist between in vitro systems and culture protocols? You can see that there were great differences between them. So different model systems grown in different protocols have different matching to in vivo development. But you can try your cells here at this website, which we have conveniently made for the field. So if you are doing this type of research and you're interested in seeing how well your cells match based on all of the algorithms we described, you can just upload your data to this website. It will run all of the processes for you, and then email you back the results, giving you, all the graphs that we showed here. Okay, so if this is a good idea, no good idea comes alone. So there was another paper that came out in Cell just after this paper, where they try to do almost the exact same thing. So what they try to do is evaluate how well iPSCs turning into any type of tissue in the human body match the human body. They did this by inputting a whole lot of expression profiles from Geo into an algorithm, a machine learning algorithm like I described previously. And what they intend to do is the same as us, where you input your reprogramming data set, conversion, differentiation, expression profile into CellNet. It gives you an output of how well your cells match in vivo developments, or in vivo different types of cells. So there are some, so I described what we have done with context so far, but I'm going to describe a little bit about what we intend to do in the future. So context, the whole pipeline, can be used as a gold standard for optimizing neuroprogenitor differentiation protocols. So if you want to add this particular type of media, or you want to add these particular growth factors, you can evaluate how well your cells match in vivo development using context and try to optimize your system. Another idea is that you can reverse engineer this to bioinformatically infer which genes are associated with maturity or regional identity. So you can try to say, this particular gene is associated with maturity, I'm going to overexpress this gene in our system, thereby creating more mature cells. Or this gene is associated with a particular regional identity, I'm going to overexpress it to generate cells of a certain type. You can also screen your cells prior to performing case control comparisons to avoid regional identity differences and ensure that you have consistent maturity. So one of the problems that I foresee is that if people are taking their induced pluripotent stem cells, and some of them are, all of the patients are forming cerebellum and all of the controls are forming cortex just by chance from the culture protocols, this would create a huge problem and huge differences between patients and controls, but that wouldn't really be true. They're just based on the culture protocols. Okay, so we want to have some methods to advance maturity. Those methods are going to rely on first bioinformatic prediction of which particular genes can change our neuroprogenitor cells to not only early neurons, immature neurons like we're getting now, but we want to change them to birth neurons or neonatal neurons, or even change them even farther to aging neurons. So if we want to model, for example, Alzheimer's disease, we want to push our neuroprogenitor to differentiate into aging neurons, advance this process. We want to do this by overexpressing genes associated with that are associated with this maturity, overexpressing microRNAs associated with this maturity, or even compounds like drugs that are associated with maturity. And we can screen using context, the system I just described, looking at morphological maturity, physiological maturity, and even epigenetic age to try to see if we can advance the maturity of our cells. One of the things that Luis has noticed in our cultures is that when we do primary human neuronal cultures, so if we take the cortical plate from our fetal tissue, so here you can see the fetal tissue, here is separated into subplate and cortical plate, and if we directly plate this cortical plate, we get neurons that have much more synaptogenic markers, implying that they're much more mature than these primary human neuroprogenitors, than if we take the neuroprogenitors, differentiate them into neurons. So this may be one clue into trying to advance the maturity of these cells. There are, of course, limitations to the context pipeline that I described. So here we focused on examining bulk heterogeneous cellular cultures that best match the milieu of the postmortem brain. Although this is a high throughput method for evaluating culture fidelity, some experiments will require homogeneous populations of cells. If an experiment requires a homogeneous population of cells, this context algorithm probably is not the best for evaluating that. So to correct that problem, we are trying to define cell types of the human fetal brain using single-cell RNA sequencing. This is a new project that's just going on in the lab. We don't have any data yet, but just to describe to you guys what we're doing. We're going to take intact fetal human brains, cell sort and dissociate them, and acquire 10,000 individual cells. We're then going to conduct single-cell RNA sequencing and try to cluster each of the different cell types into homogeneous types of cells based on transcription profiles. This will allow us to do all of the algorithms that we just talked about, but do them on the single-cell level. So then we can say how well the in vitro culture is matching at the single-cell level. And finally, we want to, an idea for the future, if we can really do this well, is to have sort of a recipe for generating stem cells. So we have two axes. We want cells of a certain maturity, and we want cells of a certain cell type to best model whatever pathological disease process we're interested in. So if we have, for example, interradial glial cells or TBR2 positive progenitors, and we want them at 19 to 24 post-conception weeks, we add the magic factor, and it gives us whatever we're interested in. So this is really, I think, the goal of the future, where the field is heading. If we're able to do this, we'll be able to model any cell type at any level of maturity and really hone in on disease-associated processes. So you can see these are just ideas, right, of transcription factors that would be associated with changes in certain time periods. And lastly, I think that this, in general, is how we should study, or my ideas for studying at least, neuropsychiatric disease in this age. So first, we want to generate, we want to get large data sets to generate a causal basis for the disease using genetic associations. So these large cohorts of patients are already happening, and right now we're already getting high evidence variants that we can use as causal factors influencing a disease. The next thing we need is cellular pathology of a disease. So this can be done mainly through postmortem studies. We can try to look through postmortem studies at the expression of the brain or at cellular level pathology. And then we can try to model them using disease in a dish models. So we get induced pluripotent stem cells or primary human neuroprogenitor cells. We optimize our culture to the particular cell type and maturity level of interest that is pathologically affected. We can engineer different mutations into that culture using new genomic engineering methods. We can try to see if we recapitulate the phenotypes associated from the cellular level pathology in a dish. And finally, we can conduct drug screening. If we can do all of these things, which is, of course, a lot of things, we will be able to model a disease in a dish, and we will be able to try to treat that disease in a dish. Okay, so all done talking to you about science. I'd like to thank so much to Luis de la Torre Ubieta, who is pictured here, as you can see. He is a great friend and a brilliant molecular biologist that I've learned so much from in the past 3.5 years. I'm grateful to have sat next to you and to do all of our projects together. A lab husband that is none can match the lab husband that I have. So thank you so much to Luis, and thank you so much to Dan for his mentorship and taking me into the lab after doing your imaging and saying I wanted to do stem cell culture. That was brave, and I hope I did an okay job. And thank you all so much for listening, and thanks to Baljit Khakh, and thanks to Dr. Scheibel. So thank you very much.
We'll take a few questions, but before we do, I just want to remind everybody to stick around. There's a special reception in Gonda on the first floor. There'll be lots of food and beer, so ask a question and go for a drink.
Astonishing, wonderful. In that clock and using the tools, is something as simplistic as kind of like ESC-derived neuroprogenitors being delayed, could that account for some of the differences? That is, if you started doing the measurements 3 weeks, 5 weeks, or 10 weeks down the road, could that potentially account for some of those differences?
Definitely. So you mean the differences between systems? Yeah, absolutely. So for example, the ES-derived cells were only differentiated from 1 to 4 weeks, right? Our primary human neuroprogenitors were differentiated from 1 to 12 weeks. So that does account for some of the differences, and we have matched up our 1 versus 4 week progenitors, the ES cell 1 versus 4 week. And actually, in that, we didn't see significant differences, at least in the transition mapping. But even with, for example, the iPSC, some of the iPSC data sets, we did see large differences even at later time periods. So if we did 1 versus 4 week and we compared to the 1 versus 6 week from iPSCs, we found even greater matching in ours, even though they're immature cultures.
Yes, really wonderful work, because so many of us are working in this field half blind, and now having at least a tool that allows us to say, okay, this is good, this is not as good, and so on and so forth. But even on the best cases that you've shown here today, I mean, there's quite a few differences genetically from these cells and what you find in brains, and brains themselves are very heterogeneous, right? So, and I think that, you know, the concern, right, sitting back and thinking about the time where whatever you find in terms of mechanism and drug and clinical trial, right, that these differences will and may make, you know, quite an impact on the ability that anything you find in these cells to find its way into the clinic. I'm sure you've thought a lot about it. I mean, it's good that you have, you know, cells that are better than others, right? Mm-hmm. I think the model system that you're using is far better than that. The cells, I agree, you have data for that. But even in the best model system you have, is it really good enough? Because, I mean, there are, you know, clear differences in genes and modules, right?
Right.
So, I mean, there's a lot of conservation, that's good, that's assuring, but there are really clear differences. So how close do we have to get so that we are successful in this game?
Yeah.
That's essentially, you know, the question that I wanted to ask.
So it's a great question. I wish I knew the exact answer. So for example, let's just do this. Even though our cells, the ones that I showed all of the matchings from, seem to have pretty good matching, you can see here that they're not really forming. These are the cells that we perform the transcriptomic matching on the left-hand side, the PHNPCs. And on the right-hand side, these are the primary neurons, right? So even this slight difference in protocol, right, is producing massive differences in synaptogenesis, right? So if we're trying to study a specific process like synaptogenesis, I think that we need to advance maturity much, much more to be able to do any kind of good drug screening on them. If, however, we're trying to study very early neuronal processes, I think we're modeling very early neuronal processes probably pretty well. I don't have a threshold, though, to say, like, this is when this works. But I think that's a great idea to, like, say, you know, this is the level of matching that we need. This is the level of matching we need to conduct a drug screen.
Question over there?
In the Khakh et al. Paper that you referenced,
yep.
Did they separate neurons from glia?
No.
Do you have glia in your culture?
Yeah, so glia, do we have glia in our culture? That's a very difficult question to answer because GFAP and glial markers also stain progenitors, right? So that's I think a difficult question to answer, and I don't really know the answer to that. Do glia appear in the in vivo data set? Yes, glia definitely appear in the in vivo data set, but probably after that 24 post-conception week level. So after 24 post-conception week, glia are formed in the human brain, and then that's the level we expect there. So, and we definitely have the problem that we're matching heterogeneous cultures from our heterogeneous culture to the heterogeneous culture of the in vivo brain. And so once glia start forming, if our cell culture is only neurons, it's not going to match well.
Beautiful work, thanks so much. One of the things that we find in genetics now is that the effect sizes of the alleles associated with the disease are very small. And in your future expectation of a patient in the dish, you say, well, we have these causal variants, and then we go to the dish and study those. I'm sure you've thought about the effect sizes and the likelihood of being able to detect variation in your cell model.
Right.
What are your thoughts on those?
So my hope, and I think this is borne out somewhat, is that genetic variation has very low effect sizes on disease phenotypes. But we know that genetic variation has very high effect sizes on, for example, EQTLs, like on expression. So a genetic variant can explain up to 50 to 60% of the variance of a single gene. No one knows how a genetic variant affects the, for example, morphology of a cell or synaptogenesis. That is something that I think we will be able to study using this culture system, but I hope that it's going to be much higher. so that we'll be able to do these type of analyses without doing 10,000 cells, which is, at this point, going to be pretty difficult.
Another question there?
Jason, were any of your labs that you called up for iPS cell comparisons, were any of them from like cerebroid or these organoid type of
No, yeah, so
systems?
all of these so far are 2D cultures. We are very interested in testing that to see if organoid cultures actually match in vivo development better. I think the Novich lab, we said that we were going to do some work with them, and I think they're trying right now to do that. That is definitely something that we're very interested in doing, but don't have any of the data yet.
We've done some with a group from Stanford.
Oh.
Using a new protocol. And they look good, but they're not, there's this maturation issue that I think fundamentally faces the field. There's something, maybe many things that are missing in terms of synaptic maturation, although, again, in those organoids, 95% of the neurons patched had, functioning spontaneous activity, which is way, way higher than anything you'd ever get in a flat culture. At a transition mapping and context, they look more mature than typical iPSCs. They look about like these.
Any other questions? Okay, well, if you'll join me once again.